Hello everyone, welcome to SOS Classroom, and today we will discuss regarding the cell structure and cell function. In previous lecture we discussed different types of anatomical terms, anatomical position and anatomical planes. And in today's lecture we discuss the cell structure and cell function. In cell structure and cell function, what are the key points we will cover in this lecture? So we will cover today history of cell, meaning of cell, definition, types of cell, basic structure of cell, cell division like mitosis and meiosis, transportation of substances across cell membrane, different types of tissue, membrane and glands. So first of all, we will discuss the history of cell. History of cell started from 1665 by Robert Hooke, who is first observed and given the name cell in 1665. While studying the cork tissue, you can see this is the cork tissue under a microscope, which is developed by the Robert Hooke. And he only saw the cell wall. You can see this is the cork tissue. And when he saw under the microscope, then he see this type of picture. So these are the small room-like structures. This is the microscope which is developed by the Robert Hooke. You can see this is the oil bottle, flame, water flask, objective, focusing screw, barrel and eyepieces. So very old microscope because of the in 1665 this type of microscope going on. In late 17th century, next scientist Antony von Leeuwenhoek improved the microscope and observe living cells like bacteria and protists. You can see this is the bacteria and these are the protists. In 1831, Robert Brown identified the nucleus within the cell. You can see this is the cell and this is the nucleus. So nucleus identified by the Robert Brown in 1831. Next, there are two scientists, Selden and Swans. There is Matthias Selden and Theodor Swans. So propose that all living things are made of cells. Means each and every living things is made by the cells. So in the plant cells, discovery done by the Matthias Selden, and in animal cells, discovery done by the Theodor Swans. Later on, 1855, Rudolf Virchow, every cell arises from another existing cells. Means if the cell is division taking on, then form the daughter cells by the parent cells. So every cell arises from the another existing cells. It is a sentence given by the Rudolf Virchow in 1855. Next, we'll discuss the meaning of the cell. The cell comes from the Latin word. The Latin word is cella. Cella means the small room or chambers. You can see these are the very small rooms or we can say chambers. So this is the smallest unit of the life. Next, discuss the definition of cell. So basically a simple definition of the cell that cell is the basic structural, functional and biological unit of the life. Structurally means different types of cells mixed together to form the tissue and tissue form the organs and organ form the system and different types of system mixed together to form the body tissue, body organism. Next, functional, so different types of cells mixed together to form the different types of functions inside the body. And biological unit of the life means different types of metabolic activity done inside your body. Next definition, it is the smallest unit of the organism that can carry out all life processes such as growth, metabolism and response to stimuli and reproduction. So these are the simple definition of the cell you can easily remember. Next, discuss the types of cells. So there are different types of cells divided based on the different types of criteria. Like there are four criteria. Criteria number 1, based on the structures, based on the structure, based on the nutrition, based on organism, and based on functions. Based on structure, there are two prokaryotic, especially in the bacterial cells, and eukaryotic in the plant cells and animal cells. Based on nutrition, autotrophic, nutrition made by cell, and heterotrophic, nutrition taken from the another sources. Based on the organism, there is plant cells and animal cells. And based on the function, there are different types of cells inside the body, like epithelial cells, which is cover and protect the surfaces of the body. Nerve cells like neurons transmit the signals like neurotransmitters. Muscle cells helps in the movements inside the body. Blood cells like RBC, WBC and platelets. RBCs help in the oxygen transport. WBC work as a defense mechanism against the bacteria and viruses or foreign particles. And platelets work as a clotting inside the body. Next, the bone cells provide the support to the body, and reproductive cells, sperm in the male and egg in the female, involved in the reproduction. So these are the different types of cells inside the body which have different types of functions. So first of all, discuss based on the structures like prokaryotic cells. Prokaryotic cells, example, bacteria and cyanobacteria. Cyanobacteria means green algae. So what are the specification of the prokaryotic cells? These are the unicellular. Means these are the cells which can make by a single cells. So bacteria makes by the single cells. There is no nucleus. It is present in nucleus. Means there is no nucleus present inside the bacterial cells. So genetic material open. There is no nuclear membrane on the genetic material. No membrane bound organelles. So there is no membrane present on the organelles which is present inside the bacteria. What is the size? Small is the 0.125 micrometer size of the bacteria and algae cells. So these are the prokaryotic cells. Next, the eukaryotic cells present in animal cells and plant cells. So what are the specification? These are the multicellular. Multicellular means the cells present more than one. Next, the true nucleus means nuclear present with the nuclear membrane. Genetic material enclosed in nucleus. Genetic material DNA, RNA present inside the nucleus. Membrane bound organelles. There are different types of organelles present in the cells like mitochondria, Golgi apparatus. So these are the membrane bound organelles. And size of the cell generally 10 to 100 micrometers. Next, based on the nutrition, that is autotrophic cells. So autotrophic cells means cell can produce their own food. You can see this is the sunlight taking the water from the earth, taking the carbon dioxide from the environment and making the food with the help of the chloroplast, because chloroplast present on the leaf, and formation of the oxygen which animal can inhale easily. Next, you can see based on the nutrition, heterotrophic cells, animal cells. So animal cells depend on the another sources. You can see these are the nutrients and nutrients taken by the cells. These are the extracellular fluid. This is the cell membrane and cytoplasm, and different types of waste material out from the cells and nutrients taken by the cells. So this is the heterotrophic cells which is present in the animal cells. Next, based on the organism, plant cells and animal cells. Next, based on the function, there are different types of functions like epithelial cells. Epithelial cells mainly cover and protect inside of the body. So this is the epithelial cells. These are the goblet cells which can secrete the mucus, and these are the cilia, and this is the functioning, means transfer the substances from the surfaces. Next is the nerve cells or neurons. So neurons helpful for the transmission of the different types of neurotransmitters inside the body. You can see this is the nerve cells and these are the muscle cells. So this is the neurotransmitters transfer inside the cells and function as a muscular contraction. Next is the muscle cells. So muscle cells help in the movement of the body, especially in the skeletal system. Next, the blood cells, RBCs, WBC, and platelets. So RBCs help for the oxygen transport, WBC for the defense mechanism against the foreign particles, and platelets for the blood clotting. Next, bone cells provide the support to the body. And last is the sperm cells and egg. Sperm cells for the male and egg cells for the female. You can see this is the sperm cells and this is the egg cells. So helpful for the reproductions. So now we discuss the basic structure of the cell. What is the general structure of the animal cells we will discuss. So you can see this is a very complicated structure of the cells. You can see. So first of all, you can see this is the nucleus. This is the chromatin, nuclear pore, nuclear envelope, nucleolus, glycogen granules. These are the cytoplasm, rough endoplasmic reticulum, ribosome, Golgi apparatus or Golgi body. You can see microtubules. These are the different types of microtubules. Mitochondria, peroxisome, smooth endoplasmic reticulum. There is no ribosome present on the ER. Lysosome, secretory vesicles, plasma membrane, centrioles. These are the centrioles. Centrosome, microvilli. These are the microvilli, microfilaments. These are all are the cytoskeleton, microtubules, microfilaments, and intermediate filaments. These are the cytoskeleton. This is the flagellum, and these are the cilia. So this is very complicated structures we will discuss in a very easy manner. You can see very simple structure of the cell. You can see this is the nucleus. Inside the nucleus there is nuclear pore, nucleus, nucleoplasm, nuclear envelope, rough endoplasmic reticulum, mitochondria, cytoplasm, secretory vesicles, Golgi apparatus, peroxisomes, centrosomes and centrioles, lysosome, ribosomes, cilia, and cell membranes. So we will discuss in a detail. So basic structure of cells mainly classification in the two forms, that is the plasma membrane and cytoplasm. Another organelles are also a nucleus, but nucleus are also present inside the cytoplasm. So you can see plasma membrane. So this is the plasma membrane and cytoplasm remaining all the contents inside the cell membrane or plasma membrane. So what are the organelles present in the cytoplasm? So cytoplasm divided into two parts, that is the cytosol and organelles. Cytosol is a gel-like fluid present in the cell and organelles. So different types of organelles present in the cells. Organelles means little organ or small organs. So nucleus, ER means endoplasmic reticulum, Golgi apparatus, mitochondria, microfilaments, microtubules, intermediate filaments, centrioles, and cellular extensions. So there are three cellular extensions we will discuss: cilia, flagellum, and microvilli. So first of all, we start the plasma membrane. Later on, we will discuss the remaining things. So what is the definition of plasma membrane? You can see plasma membrane is the outer membrane. So this is the plasma membrane, outer covering of the cells. It is a thin, flexible and semi-permeable membrane because of the transportation of the different types of substances across the cell membrane inside and outside of the cell. Cell membrane also differentiate the external environment and internal environment. External environment means extracellular fluid and cytoplasm. And what is the structure of the cell membrane is given by the Singer and Nicholson. That's why it is called fluid mosaic model. So fluid mosaic model is given by the Singer and Nicholson in 1972. According to this, what is the structure of the plasma membrane? So plasma membrane made of a phospholipid bilayer and associated proteins. Phospholipid bilayer means this is the one layer and this is the another layer, and associated protein, different types of protein present in the cell membrane. So phospholipid bilayer arranged in two layers, that is the hydrophilic head outwards and hydrophobic tails inwards. So this is the head and this is also head and this is the tail. Head made by the phosphate molecules and tail made by the fatty acids. So you can see this is the plasma membrane, phospholipid bilayer arranged in head and tail, head outside and tail inside in the outer leaflet, and inner leaflet head is inside and tail is outside. Inside the structure, the outer leaflet and inner leaflet cell membrane are not identical. Means the outer leaflet of the cells and inner leaflet of the phosphate layer are not the identical or symmetrical. These are the slightly different or asymmetrical. Outer leaflet made by the two different phosphate molecules and inner leaflet is made by the two different phospholipid molecules. Like outer leaflet made by the phosphatidyl choline, that is PE, and sphingomyelin. So these are the phosphate molecules make the outer leaflet of the head. And inside the leaflet, phosphatidyl ethanolamine and phosphatidyl serine. So these are the different mainly present in the outer leaflet and inner leaflet. Another major difference is outer leaflet attach carbohydrate, but inner leaflet don't have a carbohydrate molecules. Next, there are mainly three molecules present inside the cells like protein, lipids, and carbohydrates. So protein mainly present 50% and lipid present 47% and carbohydrate molecules present approximately 3%. So, but first of all, discuss the protein. So peripheral protein and integral protein, there are two types of protein present in the plasma membrane. So peripheral protein present on the surface and integral protein present throughout the plasma membrane. And different types of integral protein present like a channel protein, carrier protein, enzyme protein, receptor protein, and linker protein. You can see some protein inside like the channel protein. This is the channel protein. This is the integral protein, also called a carrier protein. This is the peripheral protein. Peripheral protein present both sides, outer side of the leaflet and inner side of the leaflet, but not the throughout the cells. You can see this is the glycoprotein. When the carbohydrate molecules attach with the protein, it forms the glycoprotein. When the carbohydrate molecules attach with the lipids, then is formed the glycolipids. And some of the cholesterols are also present inside the plasma membrane. And this is the enzyme proteins. This is the receptors protein. So we will discuss in the details. So this is the channel proteins. In the channel protein, this is the channel, like these are the molecules firstly attach and go into the inside the cells by the tunnel or channels. So channel protein is a form or a pore throughout the cell membrane, creating a passageway for specific ions or molecules like water to move across the membrane down their concentration gradient without the cell expending energy. Next is the carrier protein. In the carrier protein, you can see this is the tunnel protein or channel protein, and this is the carrier protein. In the carrier protein, there is no channel, there is no tunnel. Firstly, molecules attach and protein change in structures and transfer the molecules inside the cells. So this is the carrier protein. And carrier protein are the transmembrane protein that facilitate the movement of specific ions and molecules across the cell membrane by undergoing a conformational changes after binding to their target substances. Next is the enzyme protein. In the enzyme protein, you can see it is a type of protein that acts as a biological catalyst, speeding up chemical reaction in the body without being consumed by the reaction. So these are the different types of enzymes attached and different types of functions by the enzyme protein. Next is the receptors protein. In the receptor protein is a protein that acts as a receiver or transducer of signals, allowing cells to respond to chemicals messengers like hormones and neurotransmitters and drugs. So receptors protein cannot enter or receptors cannot enter inside the cells, but receptor can stimulate the signals inside the cells. You can see this is the receptors attached on the receptor proteins and give the signal inside the cells, and cell can changes different types of action. Next is the linker protein. In the linker protein has to join internal cellular structure with the external collagen or component to hold the structure in place. You can see this is the plasma membrane and this is the linker protein. A linker protein inside attached with the microfilaments or cytoskeleton like microtubules, intermediate filaments. These are the microfilaments, and outer attached with the collagen fibers. So this is the collagen fibers you can see, and this is the protein, linker protein. You can see linker protein, carbohydrate protein and some polysaccharides molecules. So linker protein help to hold the structure in place by communicating with the internal environment in cytosol or cytoplasm and external environment with the collagen fibers. Next is the carbohydrate molecules attached to the protein and form the glycoprotein. You can see this is the glycoprotein, and attached to the lipid molecules and form the glycolipid for the cell recognition. Cell recognition help in to identify the similarities of the cells and to prevent from the foreign particles. Next is the cholesterol molecules also attached inside the plasma membrane. You can see this is the cholesterol molecules. This is the cholesterol molecules. So cholesterol molecules inside the cell give the stability to the cell and flexibility in the plasma membrane. So why it is called fluid mosaic model? Because lipid and protein moves like a fluid and forming a mosaic-like pattern. You can see mosaic-like pattern. So this is the tunnel protein, and you can see this is the movement, laterally movement. This is called fluid mosaic pattern. That's why it is called fluid mosaic model. Next, discuss the function of the plasma membrane. So this is all about the plasma membrane. Now discuss the plasma membrane functions. So first function is selectively permeable. Selectively permeable means regulate the entry and exit of substances. Not all the substances can enter inside the cells. Only the selected substances can cross the cell membrane and go into the cells. For example, you can see this is the tunnel protein or channel protein. So substance directly move inside the cells, and this is the carrier protein. So only selected molecules can transfer inside the cells and can transfer outside the cells. Next is the protection. Protection from the internal components of the cells. You can see this substance transfer from the carrier protein, but these molecules cannot transfer the cell membrane, cannot cross the cell membrane because of the large in the size. This help in the protections. Next function is the communications. So membrane has receptors protein for the signaling. You can see this is the receptors, attachment of the molecules and function inside the cells. So this is the communications functions of the plasma membrane. Next function is the transportation. Transportation means the transfer of molecules inside the cells and outside of the cells. So there are two mainly transportation, that is passive transport and active transport. Passive transport means there is no need of the energy in the form of ATP, but active transport needs the energy in the form of ATP. Passive transport like diffusion, osmosis, and facilitated diffusion. And active transport require the ATP, for example, sodium-potassium pump. You can see this is the diffusion process. In the diffusion process, movement of the solute. In the osmosis, movement of the solvent. In the active transport, need the energy and movement of the sodium and potassium inside and outside of the cells. Next function is the endocytosis and exocytosis. Endocytosis and exocytosis is help for the movement of the molecules in a bulk, allow the bulk transport of the materials. When the material or substances in a bulk amount go into the cell, this is called endocytosis. And when the bulk amount of substances go outside of the cell, this is called exocytosis. You can see this is the process endocytosis, and this is the process of exocytosis. Endocytosis, the molecules or substances go inside the cell, and exocytosis, the movement of vesicles or substances from the inside to the outsides. Endocytosis process is completed in the two forms. Number one is the phagocytosis, means cell eating, and pinocytosis means cell drinking. You can see in this image, this is the phagocytosis process, especially in the case of the bacterial engulfing. You can see this is the bacteria, and cell cover the bacteria and take the bacteria inside the cell, and by the lysosome hydrolysis enzyme kill the bacteria inside the cell. So this is the process is called phagocytosis. It's the endocytosis process. This is the pinocytosis process, means small solute molecules or water substances directly taken by the cells in the form of pinocytosis when cell is drinking. Next function is the cell recognition. Cell recognition serves by the glycoprotein and glycolipids. We already discussed the glycoprotein and glycolipids present on the cell membrane. So different types of glycoprotein and glycolipids help for the recognition of the cells and prevent from the foreign particles. So this is all about the plasma membranes. Now discuss the cytoplasm. What happened in the cytoplasm? So what is cytoplasm? Cytoplasm is the entire cellular contains enclosed by a cell membrane. You can see this is the cytoplasm. Cytoplasm included cytosol and organelles, excluding the nucleus. These include both the cytosol and all the cell organelles, where the cytosol is the gel-like fluid portion of the cytoplasm. So nucleus and cell membrane, remaining all the contains is called the cytoplasm. When the organelles removed from the cell, the remaining contains or remaining gel-like fluid is called the cytosols. So cytosol also a jelly-like aqueous component of the cytoplasm, consisting of the water, ions dissolved in the molecules, and protein in the cellular organelles are suspended. So this is the nucleus and this is the plasma membrane, and remaining all the contains is called a cytosol without the organelles. Now discuss the cell organelles. So there are different types of organelles suspended in the cytoplasm. So first of all, discuss the nucleus. So nucleus is the large organelles inside the cytoplasm. What are the structures of the nucleus? You can see this is the nuclear envelope. So nuclear envelope made by the two membranes. Number one, outer membrane attached with the cytoplasm. Inner membrane attached with the nucleoplasm. This is the nucleus, this is the nucleolus, this is the chromatin or genetic materials, and this is the nucleoplasm, gel-like fluids, this is the ribosomes, and these are the nuclear pore or opening. So nucleus present in the oval shape or spherical in shape, enclosed by a nuclear envelope. This is the nuclear envelope consisting of nucleolus and chromatin. You can see this is the nucleolus and chromatin. Contain fluid is called a nucleoplasm. We already discussed this is the nucleoplasm, and formation of ribosome and exit by the nuclear pore. So ribosome formation inside the nucleolus and ribosome exit from the nucleus with the help of the nuclear pore and attach with the endoplasmic reticulum and form the rough endoplasmic reticulum. So this is the nuclear envelope. You can see it is a double layered membrane perforated with the pore which control the flow of material going in and out of the nucleus. Outer layer connected with the endoplasmic reticulum. So you can see this is the nuclear envelope, outer membrane. So this membrane directly connected with the endoplasmic reticulum and attached with the ribosome and called the rough endoplasmic reticulum. Next is the nucleoplasm. So this is the gel-like material. Fluid help in maintain the shape and transportation of molecules within the nucleus. So transportation of material means especially the transportation of the ribosome inside the nucleus. Next is the chromatin. So you can see this is the genetic material which is composed of different types of molecules, like 30% of the DNA, our genetic material, 60% of globular histone proteins, which packages and regulate the DNA, and 10% of the RNA chains. So chromatin made by the three components, 30% DNA, 60% histone protein, and 10% RNA chains. Next is the chromosome. So it is a containing with the DNA and condensed form attached to a histone protein. So you can see this is the DNA double helix in structures. This is the histone proteins, and when the helix structure attached with the histone protein and form the nucleosome. When different types of nucleosome is called a chromatin, and group of the chromatin is called a chromatin fibers. And chromatin fibers make the loop structures. When different types of loop structure make in a cluster, then it is called chromatids. This is the one chromatid and this is the another chromatid. And chromatid attached with a single point, this is called centromere. So one chromatid attached with the another chromatid with the help of the central point, that is called centromere. So this is all about the chromosome. Next is the DNA. DNA is a deoxyribonucleic acid contain the information needed for the creation of proteins, and DNA needed for also cell division and organism reproduction and health. Next is the chromosome. So chromosomes have RNA. RNA are made from the DNA templates. For example, messenger RNA in the short form mRNA direct the protein synthesis. Means mRNA give the direction for the protein synthesis. Means start the protein synthesis is message given by the messenger RNA. Next, ribosomal RNA work as a join with the ribosomal protein to make the ribosome. You can see in the images. Next, the transfer RNA bind to an amino acids and hold in place on a ribosome. So these are the different types of RNA: mRNA, rRNA, and tRNA. Next, we discuss the function of the nucleus. There are different types of functions. First is the control center of the cell, because it regulates most of the cell activities. That's why it is called control center of the cell. Next is the storage of the genetic information in the form of DNA or gene. Control of cellular activity like protein synthesis, ribosome production, and exit of the ribosome and attached with the endoplasmic reticulum. Cell growth and cell division like mitosis and meiosis, and transport regulations. So these are the main function of the nucleus. Let's discuss the endoplasmic reticulum. So there are two types of endoplasmic reticulum. So what is endoplasmic reticulum? It is a large network of membrane. You can see this is the large network of the membrane, acting like a factory and transport of system. It is of two types like rough ER and smooth ER. Rough ER when ribosome attached on it, smooth ER when ribosome don't attach on it. So what is the rough ER? Rough ER means ribosome present. You can see in the image what happened in the rough ER. This is the protein synthesis. You can see these are the protein molecules going inside the vesicles and formation of the vesicles. So this protein molecules comes from the nucleus. Protein folding and modification. You can see this is the protein folding and modification and circled by the vesicles. And transport protein to the Golgi apparatus in the form of vesicles. So this protein in the form of vesicles transferred to the Golgi apparatus. So this is the function of the rough ER, formation or modification of the protein and transfer the protein in the form of vesicles to the Golgi apparatus. Next, the smooth ER. Smooth ER means the ribosome don't present on the ER. What is the function of the smooth ER? Synthesis of the lipids, cholesterol and steroid hormones, detoxification of the drugs and harmful substances, carbohydrate metabolism, and store calcium ions and help in the muscle contraction. So basically the smooth endoplasmic reticulum work as a formation of different types of hormones, chemicals, and lipid, cholesterol, and steroid hormones. Next is the Golgi apparatus. It is also called a Golgi complex. It is also called a Golgi body. So you can see this is the structures. So it has this phase toward the nucleus side and trans phase toward the nuclear membrane sides. And Golgi body or apparatus present in the three form. Number one, in the cystine, this is the flattened sac structures. Next present in the tubules structures. So these are the tube-like structures. These are the flattened structure that is called cystine. And third is the vesicles. So this is the vesicles formations. So Golgi apparatus also called a post office or packaging center. So what is the function of the post office? Different types of packages of the material and transfer from the one place to the another place. Same like a function of the Golgi apparatus. Packaging of the vesicles, packaging of the protein and transfer to the cell membrane. So that's why it is also called a post office or packaging center. You can see these are the lumens or flattened surface, cystine, tubules and vesicles. So what are the function of the Golgi apparatus? You can see in the animation, these are the protein molecules. So protein modifications and adding the sugar, then it is called glycoprotein, and adding the lipid, then it is called glycolipid. So first function is the protein and lipid modification. Adding sugar, glycoprotein, and adding the lipid is called glycolipids. These are the modifications. Next function is the protein and lipid packaging. These are the protein and lipid packaging into a small sac that is called the vesicles. And vesicles release and transfer to the cell membrane. And cell membrane exit the vesicles toward the extracellular fluid that is called exocytosis. Next is also formation of the lysosome formations. Lysosome is a hydrolase enzyme which will help for the digest the cell when it is not working, or digest the different types of toxic material which is present inside the cells. Next is the secretions help in the exocytosis and releasing the enzymes, hormones and mucus. So these are the four functions of the Golgi body. Next organelles is the mitochondria. So you can see structure of the mitochondria. So mitochondria made by the two membrane. This is the outer membrane which is separate with the cytoplasm, and this is the inner membrane. So inner membrane present in the folding structures that is called cystine. You can see This is the cristae, inner membrane folding structure, that is called cristae. You can see outer membrane, and this is inner membrane. The space is called intermembrane space or mitochondrial space, and ribosome presents, and this is the self DNA, and this is called F0 portion and F1 portion of the epithelial complex. ATP complex. ATP complex help in the different types of formation of the Krebs cycles and chemical reactions. So mitochondria present in the oval or spherical shape, also called a sausage shape. It is a powerhouse of the cell because formation of the energy in the form of the ATP. It's a two membrane, outer membrane permeable for the transportation of molecules, and inner membrane highly permeable membrane. It has dense folding called the cristae. You can see these are the folding membrane, that's why it is called a cristae. The protein unit synthesized in the form of ATP is called as F0 and F1 by the ATPase complex. This is the ATPase complex present on the inner membrane of the mitochondria and help for the different types of chemical reactions, especially in the Krebs cycle and another also, and formation of the different types of molecules, ATP, in the as a form of energy. That's why it is called a powerhouse of the cells. Next, what are the functions? ATP productions already discussed. Next, the regulation of metabolism, like control of the breakdown of carbohydrate, fats, and proteins. Heat production, generated heat by the Krebs cycle. Calcium storage, maintain the calcium ions balance in the cells. And apoptosis, means programmed cell death. Next is the steroid and hormone synthesis. So these are the main functions of the mitochondria. Next is the microfilaments. So different types of microfilaments, or we can say cytoskeleton, present inside the cells to maintain the cell structures. So microfilaments are the thinnest filament of the cytoskeleton in a 7 nanometer in diameter, made of actin protein. Actin protein is contractile protein which will help for the movement and shape of the cell. So this is the actin protein, and you can see this structure. This is the microtubules. This is the plasma membrane. This is endoplasmic reticulum, microfilaments and intermediate filaments, and ribosomes. So these are the microfilaments. You can see these are the microfilaments which can maintain the cell structure, cell shape, and actin protein also help in the contractions of the cells. Next filament is the microtubules. So microtubules are the thickest filaments of the cytoskeleton. That is the length is diameter in 25 nanometer and made by the tubulin protein. Tubulin protein is rigid protein. Their hollow tubes are highly dynamic in structures. You can see this is the microtubules. So different types of microtubules functions, like maintain cell shape and provide cell rigidity, cell division, form the mitotic spindle with separate chromosome during the cell division. So main important role in the cell division, and formation of the cilia and flagella, and intracellular transport, and transport of the different types of molecules with the help of the microtubules. Next is the intermediate filaments. Why it is called intermediate? Because of the has 7 nanometer of diameter. Because microfilament has 7 nanometer and microtubules has 25 nanometer diameter. So it has between both them 10 nanometer diameter. What the function of the intermediate filaments? Maintain cell shape and provide the cell rigidity. You can see these are the intermediate filaments. These are the intermediate filaments. Next, discuss the centrioles. So centrioles are a cylindrical structure made of microtubules arranged in 9x3 pattern, means 9 triplet of 3. You can see this is the centrioles. This is the 1, 2, 3, 1, 2, 3, and 1, 2, 3, 1, 2, 3. Same like the 9 pairs of 3 triplets. They are usually found in the pairs inside a region called the centrosome, organization in the center. What is the function of the centrioles? Number one, cell division and formation of the spindle fibers and formation of the cilia and flagella. You can see 9:3 pattern. This is the 9:3 pattern. This is the 1, 2, 3 centrioles make the 1. Next 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Same like 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are the 9:3 patterns. Next, discuss the cellular extensions. So there are 3 cellular extensions like cilia, flagella, and microvilli. You can see these are the microvilli, these are the cilia, and this is the flagella. So cilia, just looking like a hair-like structures, mainly present on the uterus or some respiratory tract. Short projections composed of the 9 pair of microtubules surrounding a central pair. You can see this is the central pair surrounding by a 9 triplet of microtubules. Next is the function of the cellular extension, that is cilia. Movement of particles from cell surfaces. So these are the particles movement from one place to the another place. Next is the flagella. So flagella looks like a cilia, but it is longer from the cilia. For example, sperm tail in the male. So this is the male sperm and this is the flagella. So flagella help in the movement of the sperm cells. And next is the cellular extensions. So cellular extensions like microvilli. Tubular extension of the plasma membrane contain a bundle of actin filaments and function is increase surface area for the absorptions. So cilia, microvilli mainly present inside the intestine. You can see this is the structure of the intestine and these are the structures. So microvilli help for the maximum absorption of the nutrients. So this is all about the cell structure and cell physiology. Next we will discuss the cell division, different types of cell division, cell cycle, what is the cell cycle, mitosis and meiosis. We will discuss in the next lectures. So click on the next video. We will see you there. And thank you so much and thank you for watching this video. Thank you so much.